Hello, everybody, and welcome to today's webinar, Unlocking the Potential of microRNAs in Challenging Samples, Optimized Workflows for Biomarker Discovery. I am Sydney Panilla of LabRoots, and I will be your moderator for today's event. Today's web seminar is presented by LabRoots and brought to you by BioEcho Life Sciences. To learn more, visit bioecho.com. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Submit. We will answer as many of your questions as we have time for at the end of the presentation. You may also use the Ask a Question box to let us know if you happen to have any trouble seeing or hearing the presentation. Okay, I would now like to welcome our speakers for today's webinar, Matthias Hackel, PhD, CEO, CSO, TAmiRNA, and Vanessa DePasca, Sales Manager, BioEcho Life Sciences. Matthias and Vanessa, you may now begin your presentation.
Great, thank you, Sydney, for the kind introduction. Thank you, BioEcho, also for hosting this webinar and giving also me the opportunity to talk a little bit about our experiences with microRNA biomarker discovery, especially when you go a little bit off the usual path in working with cells and tissues and venture into biofluids and more challenging tissue materials such as FFPE, which is going to be the purpose of my talk today. I said my name is Matthias Hackel. I'm a CSO and co-founder of TAmiRNA. TAmiRNA, to introduce you to the company, we are a small company based in Vienna, in Austria, and for the last 12 years since the foundation, we have been really dedicating a lot of our efforts and R&D towards microRNA biomarker discovery. And this ultimately ended or led to the development of hepatomir. So hepatomir is our trade name for a set of liver microRNA biomarkers, for which we have developed a diagnostic test that has achieved regulatory approval in Europe, and that is also being taken up in routine clinical testing. I think one of the first entirely microRNA-based diagnostic tests that is used in clinics in Austria, Germany, and Switzerland at the moment. So that means overall that we have a very strong focus in the company on RNA biomarker discovery, including also validation. And besides applying this knowledge in our own R&D, we actually also offer is through a fee-for-service or contract research service business. And as said, clearly microRNA is one of the key focuses, but recently we have extended also our pipelines to extracellular mRNA, long non-coding RNAs, and circular RNAs. Minimal invasiveness is important. So a lot of our experience comes from liquid biopsies. We work with different sample types, and more recently also extending more into exosomes and extracellular vesicles, although I won't be covering a lot of this today. And then I will also touch a little bit about our experience with tissues and especially FFPE. But first of all, I just want to highlight 3 reasons why we as a company sort of, trust in microRNAs, if you want, and have decided to work with these molecules as our, biomarker targets. And first of all, and this actually also, last year has been acknowledged with the Nobel Prize. MicroRNAs are quite unique in the way that they shape the transcriptome and regulate gene expression. And I mean, this is a very simple visualization that you can see on this slide. Essentially, it shows that a single microRNA based on the hybridization with an mRNA can inhibit its transcription. But what's interesting is the multiplicity. So that means that a single microRNA can have interactions with multiple mRNAs that may result in complete or incomplete inhibition. But that means that individual microRNAs can have quite a broad impact on the transcriptome. And that also enables just 2,500 microRNAs to regulate 70% of the human protein coding genes. So they're biologically highly relevant. Going to the next slide. The next hallmark that we think is really important from a biomarker point of view is tissue specificity. I hope you can see the slide. For me, it's not showing yet. There it is. So high tissue specificity is, especially when you work with minimal invasive detection in, liquid biopsy, it's always good to understand where the biomarker is coming from. And that is enabled by tissue specificity. And although not all microRNAs are tissue specific, there is quite a large amount of microRNAs that you find transcribed in many different cell types. There are several, and about 10% of microRNAs do show very, very specific transcriptional patterns or are restricted to specific cell types. You can see here on this slide, there from two papers, representative figures. On the left is from a rat study where the authors have investigated microRNA transcription about 20 different tissues from rat. And in this heat map, you can see on the right side, especially several microRNAs that give a signal in the form of a red color in this heat map only in isolated tissue types, such as, for instance, the pancreas for microRNA 216 or 217, the liver for microRNA 122, for example, or the muscle for microRNA 1 and 133. So these microRNAs fulfill really important functions in these tissues to maintain the sort of the specialized cell function of these cell types. And what's interesting is that this tissue specificity is highly conserved. The same is true for human samples, but on the right side, you can see in situ hybridizations of specific microRNAs in zebrafish, and you can see that also here microRNA 122, for instance, you can see the signal is restricted to the liver in zebrafish. So, so these is highly conserved across species. And the final reason or attribute of microRNAs that is really important in our view as a biomarker is minimal invasive detection. So we can measure microRNAs from biofluids, and essentially there are two mechanisms that enable us to do that, or that are behind the release. And this is on the one hand, passive release shown on the left side. It's a very simple mechanism. This is in an acute setting, acute injury or acute disease where cells become necrotic. You have a leaky plasma membrane of the cells, and that enables amongst others microRNAs to leak into the supernatant and ultimately into circulation, and there they're stabilized by the incorporation in protein complexes. So microRNAs are really locked into the so-called RISC complex. This is a very very tight interaction, and this also stabilizes microRNAs for quite some time in the extracellular space. In contrast to passive release, actually what is happening constantly in our bodies is active release of microRNAs, and there are different mechanisms behind this. There's shuffling into larger, smaller vesicles, or also release associated with proteins. And this is a physiologic process believed also to enable cell-cell communication. But it has been shown that this active release changes or can change with the onset and progression of disease, which is for us relevant to utilize them as biomarkers. So how do we develop microRNA biomarkers in the company? This is shown on this next slide, which is it's a fairly busy table, but I'll quickly walk you through. So essentially what this slide shows are the different stages that we go through and that we have to pass when we go from an idea. Here on the side, you have the planning phase. This is where you start with the idea of a new biomarker, so-called target product profile, can be useful to structure this approach. And then from this idea, you work towards the right, which is product that shows clinical utility and shows a certain performance in diagnosing a disease or predicting an outcome that is clinically useful. Right, you have to go through different stages. Clearly, samples are very important. I'm not going into detail on different pre-analytical attributes, although this is a very important topic. But for my part, I really focus on the second step in the process after you have recruited the sample, which is biomarker discovery, as is really the first important stop-go decision point in biomarker development. So biomarker discovery in terms of microRNAs or general cell-free RNA is a bit challenging. And the reason for this is that I always put it like this: extracellular RNA is different. And I'll try to explain to you what I mean by that. So I'll start with not extracellular, but intracellular RNA. So here you can see a so-called electropherogram from a method called the bioanalyzer. or nowadays also fragment analyzer, different instruments that can generate this analysis. Well, essentially what you see in this slide is the data for one RNA preparation, and on the x-axis you see the RNA length in nucleotides from the low molecular weight standard that is indicated up to the ribosomal RNA 18S and 28S subunits that give very distinct peaks, and these are also highlighted here. So essentially, this is a very high quality RNA sample. You can see it's about 100 nanogram per microliter. It comes from brain tissue, which has very rich transcriptome. And based on this RNA, you can apply different metrics. You can apply an RNA integrity number, or RIN, which is shown in this little box here. You can apply the DV200, the fraction of RNAs that are greater than 200. Or you can also analyze such an RNA definitely by spectrophotometric analysis, such as a nanodrop, and have purity estimates based on 260 to 80 or 260 to 30 ratios from that. So that is not a big challenge. However, when we go and we show the same profile now for an extracellular RNA, you can clearly see it's different. You can see that the ribosomal RNA peaks are gone, so we cannot really estimate an RNA integrity. You can see the low molecular weight standard appears now very large, and this is because concentrations of extracellular RNA, if we work with smaller volumes, are very low, usually below 1 nanogram per microliter. And we can really only see that there's sort of an enrichment of smaller fragments starting with around 20 nucleotides to maybe about 500 nucleotides. And so the QC of these types of RNAs and to take decision based on such analysis is very challenging, and actually not really, in our experience, not really meaningful. So that's why with our cell-free RNA or microRNA biomarker discovery workflows, we rely on a different type of QC of these samples. And I'd like to talk a little bit more about this. So what you can see on this slide is the workflow for our biomarker discovery assay. It's a small RNA sequencing based assay, meaning a next generation sequencing, so untargeted. Can measure hundreds to up to 1,000 microRNAs with this assay. It starts with an RNA extraction, and then usually workflow you go to library preparation and sequencing. However, due to the challenge that I just explained to you, we have added a step to that. And the step that we have added is that we spike, meaning that we add synthetic oligonucleotides to each RNA sample that we process. These are called MIND spike-ins that span a certain concentration range that overlaps with endogenous RNAs, and which are really insightful for us then to assess the quality of the RNA and potentially also to normalize it. And this is described in detail in this publication that you see there highlighted on the right. So to explain a little bit more in detail about these spike-ins, the MIND spike-ins, as I said, there are 7 of them that we are using. They are each distinguished from each other based on a unique core sequence that has 13 nucleotides. You can see those cores highlighted here also in the table. And these cores, by the way, are not found in nature. So we screened them against human, mouse, rat, but actually virtually all mammalian species to make sure that there's no, if we add them to a sample, there could not be any interference from a residual or background presence in nature, essentially. And then you can see that the special feature of these spike-ins is that the core sequence is flanked on the 5' and 3' end by random positions, in total 8 random nucleotides, 4 on the 5', 4 on the 3' end. And that actually increases the diversity for each spike-in enormously. So if you do the math, you have around 65,000 different variants per core sequence. And this actually is crucial to prevent any sequencing or ligation bias in the assay. And so that we have in our sequencing data, we have a true representation of this abundance. And this is what you can see here in the plot here on this slide on the left side. This is actual data from a small RNA sequencing run for a specific sample. And what we do is we recover the raw read count for these spike-ins. You can see this on the x-axis. And we plot the sort of observed or measured read count with the intended concentration, so the copy number on the y-axis. We check the fit, and we can thereby also control the dynamic range of the assay. And then importantly, we can then relate the range of the spike-ins with the dynamic range that we see in the microRNAs that we detect. And this is shown in this middle part, where you have a lot of dots. The blue dots and the red dots are all microRNAs. The blue dots are different microRNAs that are in range. That means they are within the between the lower limit and the upper limit of the spike-ins. And then you can see there are also microRNAs that are below the lower limit or above the upper limit. And this really informs us about the RNA quality, because we do expect for all high quality extracellular RNA samples that there's a really decent overlap and high dynamic range in the endogenous microRNAs. What it also enables us to do and is really to confirm if our sequencing method has low ligation and sequencing bias. And you can see that we can look at individual spike-ins, and then we can, what you can see here on this in this figure is the composition of a spike-in at each position in its length. And when you start with the middle part, you can see the core sequence, which we have clearly defined. There's no variation, right? I mean, it's A, C, G, A, T, C, etc. But you can see that the first four and the last four positions are really a more or less balanced composition of all four nucleotides, which shows that this sequencing assay has really no preference or no bias for any RNA motif or any microRNA sequence, which is what we want to have. In addition to that, there's sort of this quality control aspect. There is another way how we can utilize the spike-ins. Just wait for the slide to show up on my screen here. Yeah. And this is normalization. So normally when you have sequencing data and what we're normalizing to is sequencing depth, and this is done based on so-called reads per million. For microRNAs, if you have longer transcripts, such as in mRNA or whole transcriptome sequencing, you may do reads per kilobase and million, so you have to adjust for the transcript length. But ultimately, every target sequence shown here in red, you normalize to the total number of sequences that you have in your sample. And this ultimately assumes that the total RNA composition is actually constant between all samples. If we have spike-ins with known molar concentrations, it actually gives us another opportunity. We can actually, and this is shown now in this lower left part, you have these purple sequences, which is a fixed amount of spike-in that is now present in every sample. And so this enables us to actually exclude or sort of ignore a bit the total RNA composition, overall RNA composition, just reference a target microRNA sequence shown in red to the sequences or the abundance of the spike-ins. And this gives us more an absolute quantity that we can derive from these data sets. And I'll show you later an example where this has been really useful for us. So we have quite a lot of experience with this assay. Overall, we are probably about 5,000 to 6,000 samples that we have sequenced by now. And as you can see here in this histogram, most of them have been biofluids, also from different species. And our experience from this, especially in the biofluid sector, is that the microRNA repertoire in different biofluids is really diverse. And you can see here a simple analysis that we did with all this data is just to plot the number of detected microRNAs against the sequencing depth. And you can see the plasma and serum are fairly complex biofluids. They contain a lot of different microRNAs, while, for instance, synovial fluid from a joint space of the hip or the knee, or urine, are biofluids that contain a more specific set of microRNAs that are derived most likely, from the cell types and the tissues that are in close proximity with these biofluids, which can have an advantage. Can improve your signal to noise ratio ultimately. And finally, also to show you that while we're adding something to the library, we're not losing a lot of sequencing information. And this is shown here by the percentage of total reads from our sequencing data that actually map to the spike-ins, because we do not want to spend an excessive amount of our money and data on the spike-ins, just as much that we can learn from them. And you can see here we've optimized this, that on average, we spend about less than 1% of our sequencing data to these spike-ins. And you can see here the median for biofluids, extracellular vesicles, tissues, and cells shown here on project level. So this is actually well below 1%. So I'll proceed to a short case study on where we used this technology, and this was on CNS, so central nervous system toxicity biomarker discovery, as part of an EU-funded project called the TransBioLine project. And the underlying hypothesis or the aim of this study, that we were interested in seeing, okay, could we apply microRNAs as biomarkers or indicators of central nervous system toxicity? So if you have damage due to drug exposure or trauma to brain tissue, could microRNAs in the peripheral blood indicate that? And for us, the hypothesis was that such microRNAs should have a high concentration in the central nervous system, including cerebrospinal fluid that you can see here, and a low concentration in the peripheral blood. And only if you have damage and leakage through either the CSF blood barrier, you would see an increase of these microRNAs in the peripheral blood. So to test this hypothesis, we set up a study. This is the study design here, where we analyzed matched serum and cerebrospinal fluid, so CSF samples from two different cohorts, one with a patient cohort with multiple sclerosis and another cohort with NCD. These are non-confirmed CNS disease. So these were patients where CSF was collected, but ultimately no disease was detected. And we had matched serum and CSF samples from those, and then we sequenced the microRNAs in both, and then we tried to understand, are there microRNAs that are highly enriched in CSF compared to serum that could serve as potential biomarkers of CNS toxicity. But the first thing that we realized in doing that is that the RNA composition between serum and cerebrospinal fluid is very different. And this is what the plot on the left side here shows. And you can focus on the red bar. The red corresponds to the percentage of reads that are microRNAs. You can see here in this study in serum, we had about 25 to 50% in serum of reads mapping to microRNAs, and you can see in CSF, this is significantly lower. So the CSF composition is different, and we have a much lower microRNA part in there. But if you would utilize conventional normalization, such as reads per million, you would actually ignore that. Yeah. On the right side, you can also see that, again, the complexity in serum to CSF in the number of detected microRNAs is very different. So when we analyze the data and we focus on the absolute microRNA concentrations that we estimated from the MindSpike-in normalization, we get a very clear picture in this heat map. And this picture tells us that, first of all, based on the clustering, you can see CSF in purple and serum in green, and you can see they cluster completely separate from each other. And what you can appreciate is that in serum, you have a very large cluster of microRNAs that are high abundant and almost not present in CSF on absolute level. There's actually only another small cluster of microRNAs highlighted here by this red arrow on top, where you can see they give brighter signals across all CSF samples, but no or very low signal in serum. And these are the interesting candidates for us. These are microRNAs that apparently are brain specific or CSF specific. One example for them is microRNA 124. microRNA 24 is in terms of transcription restricted to neurons. It shows about 100-fold increase or in ratio in its CSF concentration to serum concentration, as we derived from the NGS data. And you can see in this plot, this is about the same or similar for MS and NCD patients. But what's interesting here, what I highlight, is that when we validated this by qPCR, using absolute quantification by qPCR, we get more or less the same enrichment that we get from the NGS data when we use the copy number. But you can see when we use the NGS data and the RPM, which is the dimensional normalization, we really would have overestimated the enrichment. You can see that the yellow bar for both is really off, and this shows that actually, in terms of qPCR, our observation is that the copy number-based normalization of the NGS data is more close to the qPCR results. And then for this microRNA, we went further, and we were lucky to get samples from a collaborator in the US, where they had treated rats with different CNS and nervous system toxicants, such as chloropropionic acid. And we got the serum samples from these animals at baseline and after treatment. And then we measured microRNA 124 in these samples. And you can see that on the left side, that after exposure, we see significant upregulation in the plasma or in the serum in rats of this microRNA, so indicating the toxicity. And to put this into perspective, we correlated actually the observed fold changes of microRNA 124 to neurofilament light, or NFL. NFL is sort of I think approaching or appearing as a new gold standard biomarker to assess CNS diseases and also CNS toxicities. But what we see extremely high correlation, even almost higher sensitivity of microRNA 124 to indicate histopathologically relevant damage. So this was a very good result for us. I would like to close my part here with just a view on tissue samples and potential relevance of tissue samples in biomarker discovery, as I've only talked about biofluids so far. So why do we happen to use tissue samples quite frequently actually in biomarker discovery? And there is a few advantages, or there's a few use cases, which is, first of all, it's really the higher signal to noise ratio in tissues compared to liquid biopsies. When you look on the right side, this biorender based visualization of a blood vessel, and the factors that circulate in blood, you can appreciate that there is probably a complex mixture of microRNAs secreted from a lot of different cell types, primarily blood cells and endothelial cells, but also highly vascularized tissues. And if you have only a localized change in a tissue, you probably can expect that this effect on tissue level will be diluted on the systemic level. And that means that the effect size or signal to noise ratio in the biofluid will always be lower compared to tissue. So for that reason, we have done this a couple of times, that although ultimately we want to work minimally invasively, we start discovery on tissue level, so that we really understand effect sizes and the changes on tissue level of microRNAs, and then only select some or add data on biofluids to this to relate the two to each other, or have context of understanding changes in biofluids if we have data from tissues. And this has been really in the past. The second is that we can also do it after we have done analysis of biofluids, just really to confirm the cellular origin of a signal or an RNA or circulating RNA biomarker, and really confirm that also on tissue level we see a response. It can ultimately, also working with tissues, help us to understand mechanisms of action for biomarkers, and there can also be sometimes the case that actually, we don't necessarily have to work non or minimal invasive. As some diseases, there is routinely biopsy material available as it's part of the standard of care. And then actually we can rely on tissues for biomarker development, which for the reasons above can also have advantages. So for that reasons, it was also important for us to test our workflow with such tissue-based samples. And, ideally, there's cryopreserved tissue. But our experience is that this is not always available. And in clinical studies, it's much more common, or with biobanks, that there is so-called formalin-fixed and paraffin-embedded tissue blocks that are available, as shown here on the right. But the challenge with FFP, as you may know, is that these really do not preserve RNA well, and you have highly degraded RNA. So for us, the aim was here really to test the feasibility of using and analyzing FFPE tissue microRNA using the MIND spiking, or the workflow that I've shown to you before. And not really knowing, how to best extract RNA from FFP, we did a pilot where we selected 3 different protocols for RNA extraction from FFP samples, including a protocol from Beckman Coulter called the FormaPure XL, the mRNA FFP kit, I think quite a famous kit from QIAGEN, and then also the EchoLUTION FFPE RNA kit from BioEcho. And we applied these 3 kits to 3 different FFP tissue samples, so all 3 methods with all 3 samples, and tried to learn, if there's any difference or any impact of RNA action on the results. And so the first level of quality control or analysis that it was really just looking at the RNA concentration and RNA quality. And this is shown here on the left as the fragment analyzer profiles of the RNA. And for each, I'm not going into detail here, maybe just show that the RNA concentration or the yield was comparable. But what we observed is that when you look at the at the profiles, you can see that with pure method, on which we had the lowest size of RNA, while with mRNAs and especially EchoLUTION, we've seen that the overall size range and the integrity of the fragments, that RNA fragments that we get, seem to be the longest, which is certainly a positive thing. The next level of quality control that we did with each method here was the library yield. So when we apply these RNAs and the same concentration of RNAs into our library prep, how much yield do we have ultimately for sequencing? This is shown above here in so-called again, fragment analyzer smears of the library. I'm not going into details here on this either, just to mention that the total library concentration was lowest for the FormaPure and higher for the EchoLUTION and the mRNA-easy. And what for us was important to see also that there's low variability, and we have seen that the lowest variability in the library prep we've seen with the EchoLUTION protocol. If you look to the top right electropherograms of EchoLUTION, you can also see that the library profiles are very, very consistent and similar for this method. And then what does the actual sequencing data when we performed small RNA sequencing on the samples tell us? What you can see here in this plot is again the RNA composition. You can see in red microRNAs, in green tRNAs, and you can see a fairly large bar which is ribosomal RNA. And ribosomal RNA is highly present in these samples because of the RNA fragmentation. And you have ribosomal RNA fragments in your sample, and these short fragments just spill into your size range for small RNA sequencing. So actually, we want to have this blue part as small as possible, and this red part being microRNAs as big as possible. And we analyze these, and we analyze the microRNA content. We do see that with two protocols, mRNA-easy and the EchoLUTION, we have the highest percentage of microRNA to total reads, which is important for us. And vice versa, we have a significantly lower ribosomal RNA content with these two protocols compared to the FormaPure, which is important for us. Then what did we observe in terms of RNA complexity or microRNA detection? This is shown in this upset plot. Maybe let's start with the upset plot and the bars on the lower left side. You can see the microRNAs that we detect per sample. The samples are color-coded, so green is BioEcho, red is QIAGEN mRNA-easy, and blue is FormaPure. And we can see the highest detection rates in most of the BioEcho samples, followed by QIAGEN, mRNA-easy, and then the FormaPure. Overall, with this workflow, we get around 600 to 800 microns with the BioEcho flow. And you can see in the spike-ins that on plot here with the isolation, while the right is FormaPure, you can see first of all the higher recovery of microRNAs, but you can also see nicely that we really extend here the full dynamic range of our spike-ins, which means that from these FFPE tissues, we really can recover a full dynamic range in microRNA transcription with this protocol. Right. And this is complexity in terms of the profile or the patterns themselves. We, of course, ask the question, okay, does the RNA extraction protocol impact not only maybe the number of microRNAs we can detect, but actually also the profile? This is shown in this heat map and correlation, which is actually a correlation plot. It's a bit difficult to read maybe, but just to walk you through, what we see here is that for one FFPE sample, FFPE24, we actually see a very good overlap in the microRNA profile between the three different isolation procedures. But we also see that for the two other FFPE samples, we see that each method, EchoLUTION, mRNA-easy, and FormaPure, result in a rather distinct microRNA profile shown on distant clustering of these samples. And especially, I would say, while EchoLUTION and mRNA-easy are quite similar to each other, the FormaPure for two samples resulted in a very different microRNA profile, which is a bit of a concern here. So RNA, or the conclusion here is that really the RNA protocol can actually also have an impact on not only the sensitivity of the method, but also the conclusions that we can derive from that. Yeah, so we made a few important learnings from this, and to really summarize now my part on microRNA biomarker discovery using NGS in challenging samples. It's that the workflow that we've developed and that we can also offer as on a service level is applicable from cells to tissues to liquid biopsies. And although I haven't shown that data, you can believe me, also extracellular vesicles. Although especially for liquid biopsy and tissues such as FFPE, while this is feasible, we also learned that there are certain challenges and that we really have to optimize our quality controls, our normalization procedures, that we can get high quality data from these samples and also draw the right conclusions. And for FFP tissue, especially, we realized that really optimizing the total RNA extraction protocol can have a large impact on the data. With that, I'm at the end of my presentation. I hope this was useful for the audience. I'm looking forward to the questions at the end of the session, and I'll hand over now to Vanessa to explain in a little bit more detail how the EchoLUTION protocols work.
Yeah, thank you, Matthias, for providing this exciting insight into microRNAs and your solutions for analysis. I actually just want to step in with a few slides showing you how BioEcho can actually contribute to your successful RNA analysis. If you haven't heard about BioEcho, just a quick introduction. We've been around for a while, founded by two former seniors from QIAGEN who thought they definitely needed a change in their technology and how extraction is being done. And since the end of last year, we're also part of Mera Health, which is a US-based NGS sequencing company. So I just wanted to bring closer a little bit our EchoLUTION technology that Matthias also tested for his protocol for FFPE. In summary, what we try to do is just take all the different methods that are available and just remove the pain points. So that includes especially the washing steps that you have, and just make things more convenient, faster, reduced also consumables, being more economical for costs and the environment, but also ultimately keeping the same quality and performance as with other kits. How does it work? So you basically just take your sample. Every protocol, depending on the sample type that you have, has some kind of sample lysis step. And then you would just transfer your sample that would contain nucleic acid with all kind of different debris and solvents onto our matrix. So that's the purification matrix. It's available in spill columns. We also have plates. The easy step here is now all you need to do is top it up, place it on a centrifuge, quick step, 1 minute, 1000 G, and you'll have the clean nucleic acids just in your eluate. So there are no washing steps included. Everything that you don't need gets stuck in the purification matrix. To show that, I just wanted to highlight a few things about our specific FFPE DNA RNA kit. They're fairly similar, so I'll explain the differences here a little bit later. And just the way we structured the protocol and we aligned some of the steps within there, we can shorten the time dramatically. So we're really talking about saving an hour on processing, for example, 12 samples at once. And just to show you the protocol, fairly simple. It's literally just 6 different steps. It starts as most protocols would do. You take your FFPE block, you use your slices, and we actually start with the FFPE decross-linking in the first step. And the specialty here is that we combine the tissue lysis and the deparaffinization in just one single step. From there, what you basically do is you'll have to condition our spin column. So the purification matrix always has to stay wet, so it's buffered. It's just a minute in the centrifuge, just describe the flow through. And once this is done, you can then high centrifuge the the RNA preparation of your FFPE slides and just pipette the lower aqueous phase onto our matrix, and then do another quick centrifugation step to then have the eluate. The specialty here is that our matrix is not super selective in terms of which types of nucleic acids are passed. So the actual selection happens by adding a nuclease. At the end, this means if you want to have both, you can literally also just extract DNA and RNA in one single step, or you would add a DNase or respectively an RNase, depending on which of the nucleic acids you want to continue working with. So just showing you a few of the results. You already saw a few traces from Matthias. So we have seen similar things in the past. This has been provided to us by another customer that tested our kits with another silica-based method on brain tumor samples. And here again, we see like the similarity in the trace where we just have these much longer fragments that we keep, so just resulting in a higher DV200 for the RNA extracted. And we also seeing that across all different kind of tissues. So we tested FFPE samples with brain, with kidney, with breast, with lung. So we definitely see everywhere very consistent and high yields for RNA extraction. So last but not least, also our results for the NGS analysis of the different RNAs that we extracted. So we compared again here just our EchoLUTION technology to, I would say, the standard that's being used, the silica extraction. And we can also see that it doesn't only make a difference for microRNAs, but in all RNAs in total. So we see a much higher count of different RNAs, as as well as more unique mappers in the sequencing data. So that was all I had to add. Thank you. And giving back to Sydney and open for the next part of the webinar.
Perfect. All right. Thank you both for your very informative presentation. We will now go ahead and move into the Q&A portion of the webinar. If you happen to have a question that you would like to ask, please do so now. Just type your question into the Ask a Question box and click Submit. We will answer as many of your questions as we have time for. Okay, let's go ahead and get started here. The first question that we have says, very interesting technique and rigorous work. Thank you for sharing. I just have a fundamental question. Is your protocol able to detect novel microRNAs, or are you always relying on annotations? Relevant to that, has the full microRNA landscape been sufficiently annotated, or do you see this as a limitation?
Yeah, that's to me. So thanks a lot for question. So yes, our workflow can also be, or the data workflow can also use to predict novel microRNAs that have not been annotated in repositories such as mirbase or RNA Central. And this is, the library targeted. So there's a distribution, selection steps here, will ligate between the sequencing adapters. And of course, then we, initially annotate from that data that we get microRNAs that have already been annotated using repositories such as mirbase, but there's always sequences left that we cannot annotate. And these we can run through prediction algorithms that map these microRNAs against the genome and look for, RNA folding structures. So yes, that can be done. Also with our data. And it's something that I haven't told you, something that we also do actually is looking into isoforms. So I didn't go into that, but you may know that there's some variability in the processing or in the maturation of microRNAs, resulting in 3' or 5' variations or even polymorphisms. And we also evaluate this. And if we have a biomarker of interest, we always try to understand how uniform is the effect, meaning the fold change in disease versus control across all isoforms. This is for us important to understand if we go, on with qPCR, for instance. Do we have a concern if the qPCR is isoform specific? Just to mention that. Coming to the next part of the question, do I feel that the annotation is complete? So I would say that for human, we probably are close to having a really complete picture. I mean, and the reliability of new predictions, etc., is maybe a bit challenging. Let's put it like this. But there are a lot of species. We work a lot with mouse and rat, but even pigs, chickens. So there are more model organisms, and there is definitely in these. I think we underestimate currently based on annotations that we find in miRBase or RNA Sensible, we actually underestimate the complexity of microRNAs. So I think especially if we work with non-human, the ability to predict new microRNAs is definitely important. I hope that answered the question.
All right, fantastic. The next question that we have here says, is there an EchoLUTION RNA kit for plasma samples? Does it enrich for microRNAs or total RNA?
The quick answer is no. The longer one is we are currently working on a specific kit for plasma, for blood samples. There are certain limitations to our technologies, and that is mainly that it's difficult to concentrate samples. So basically, the volume you put on top of the matrix is also the volume of your alloid. So if you want to continue with specific downstream applications and you need a certain concentration, it will get difficult if the input is already lower concentrated. So, but yeah, just stay tuned.
Yeah, let us know.
Perfect. The next question that we have here says, do you have a DOI PMID link to your paper with information on the spike-ins?
Yes, if you go to the TAmiRNA website, www.tamirna.com, you'll have a section on the spike-ins, and then you'll find also a section on the small RNA sequencing, and you'll find links to actually a couple of papers, or the original paper where we describe sort of how we develop these spike-ins, and then a lot of application papers. Yeah. You can also send us an email if you have follow-up questions on that. But the website should be fairly a comprehensive resource of information.
Perfect. The next question we have here says, as you said in case study, can we do the same for other types of cancers like breast cancer diagnosis? And based on the microRNA concentration, can we quantify the degree of the diseases or damage to the body?
So we have no experience with this indication. I mean, TAmiRNA, I didn't show our pipeline. We exclusively focus on age-associated diseases, and we have no programming in cancer. However, I mean, I think there's evidence in literature that different types of cancer, although these are localized changes in tissues, with malignant cells proliferating and growing there, that this has an impact. So not only has, of course, changes on tissue level, but also changes in then the circulation. Yeah, the mechanisms behind this, I do think it's an overlay of passive and active release. I think what can happen in a cancer tissue is that you have more excessive cell death, based, the immune system interacting with the cancer, and this is maybe something that can alter the microRNA profile in the circulation, or a mechanism contributing to that. So passive release. And in addition, I'm pretty convinced that the secretome itself in sort of this pathophysiologic condition of a cancer cell is different to the corresponding quiescent or normal cell, and this is in this altered active release of microRNA. So I think both mechanisms overlay with cancer, and I think the ability to develop a test that really works well really depends on a lot of factors. I think what certainly helps is if the tumor or the tissues is more vascularized, so you have a lot of transfer of the secretome of cancer into the biofluid, rather if it's not. But again, we have actually no experience with that in-house in our R&D that I could comment on.
Perfect. Okay, it looks like we have time for a few more questions here. This next one says, have any biomarkers identified through this workflow been clinically validated or entered translational pipelines?
I'm very happy about this question, because this is one of the big successes of TAmiRNA, that we actually have developed what we call HepatoMIR. HepatoMIR is a liver microRNA biomarker panel. We got the CE-IVD regulatory approval 2 years ago for that test, and it's clinically used now. It's used in Austria, Germany, and Swiss hospitals at the moment, and it's used specifically if you have if you undergo liver surgery, because either you have a tumor in the liver, or you maybe have an infection in the liver, and you either have to undergo what is called liver resection or liver transplantation. The test determines the risk of post-operative liver failure or morbidity. So it tries to stratify patients into those groups that will benefit from the surgical treatment and those that won't, and those who won't will undergo other types of non-surgical treatments. So yes, and this signature has been discovered using the exact workflow that I demonstrated today. You can find more about HepatoMIR also on our website if you're interested.
Okay, great. Next question says, we are dealing with urine-driven exosomal microRNAs, and we usually get low concentrations. Is there a way to enrich these microRNAs, and how can we be sure our microRNA is not contaminated with larger RNAs or other contaminants?
So we have made, you already mentioned you're working with exosomal RNA from urine. We have made the experience also working with urine, and we compare total RNA extraction from urine. You take a certain volume and you may concentrate this using ultrafiltration and to RNA extraction. And we compared this to doing prior exosome or EV enrichment from urine, and we see that the RNA associated with the EVs or exosomes seems to be less prone to, or of higher quality. Let's put it like this. Yeah. While we do see, especially in urine, also compared to plasma, there are a lot of fragments. This is less, applies less to exosomal RNA. So I think that you're doing the exosome enrichment is already good. I would recommend this for urine RNA biomarker analysis. To further enrich, so we don't do any, also in the RNA extraction procedure, we don't do a microRNA enrichment. And this is for the simple reason that either the qPCR that we use downstream of sequencing, or the sequencing itself, is quite specific to short RNAs, and we get a decent readout or output, let's put this, of microRNAs from such urinary EV samples with our workflow. So we don't see or feel that there's really a need to enrich. And although these concentrations are low, so also I showed an electropherogram from plasma with extracellular RNA with less than 1 nanogram per microliter. For urine, it's absolutely the same. So very low outputs, but if you have a sensitive detection method, this is absolutely fine. I hope this was useful.
Perfect. Vanessa, did you have anything that you wanted to add to that?
Yeah, kind of the same as Matthias was saying. It may also depend on the exact downstream application. So I feel like when it's getting complicated, it's sometimes easier to do it downstream, like on the data analysis side, than try to experimentally enrich the microRNAs. They may just introduce a lot of different biases you don't want to get.
All right, great.
Yeah, and maybe to add to that, what we do in our library prep, at the end, we do a size purification of the libraries. This is very technical, but ultimately what it means is that because also the question raised a bit of a concern about the presence of fragments that are not related to microRNAs in our sequencing data. And yes, we see this. You have seen the ribosome RNA fragments in the FFPE data, but still we try to minimize this by having a fairly narrow size range between about 16 and 36 nucleotides. that we purify before going into sequence. And for this, we are not so much worried about longer RNA fragments, because we would not see, or we actually do not see them in our data.
Okay, fantastic. It looks like we have time here for one more question, so we'll go ahead and wrap up with this one. It says, how translatable are microRNA biomarkers identified from FFPE samples in terms of diagnostic value?
That's a good question. So we have not gone down that route, and really having a development program in the company that relies on FFP and do an FFP discovery and then see, can we translate this further on. But out of experience, we have generated really good data sets for FFP tissues, mainly as part of our services. And based on the effect sizes that we have seen there, and also then, what we did do though is that we then, in a first step, try to reproduce NGS data on FFPE by qPCR. And there we have seen that there is a good overlap for microRNAs that show high effects of maybe about twofold to or greater than twofold change. I mean, the translatability, and I can spend hours on this, depends obviously on a lot of things, ultimately. But what I would look at initially, whether it's FFP, always on the effect size. So we always go for biomarkers that show in your initial data set big differences, because as you go and try to translate these into further validations into product, and you include more and more sample, meaning more and more variability ultimately, because your cohorts become larger, your effect size always drop. And if you don't have a big effect initially, you probably won't be able to increase it later on. So I think, I think that's the general challenge in biomarker development.
All right, perfect. Well, thank you again, Matthias and Vanessa, for your time today and for your very important research. We would also like to thank LabRoots and our sponsor, BioEcho Life Sciences, for underwriting today's educational webcast. Before we go, I would like to thank the audience for joining us today and for their very interesting questions. Questions that we did not have time for and those submitted during the on-demand period will be addressed by the speakers via the contact information you provided at the time of registration. This webinar can be viewed on demand. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, everybody, goodbye.
Bye-bye.